Let me start with first principles. In essence, epidemiology is a population science that makes inferences based on analyzing population distributions of health-related phenomena. The work necessarily entails comparing populations, whether at a given moment of time or over time, and this in turn requires asking who and what defines these populations, a question that is surprisingly rarely asked in epidemiology, not to mention most other population sciences. The dominant view, expressed by the figure to the right, is that populations have the properties they do because of the traits of their individuals, which in turn allows the populations to be objects of statistical inquiry, whose individuals can be sampled to give insights into the whole. The figure shown is that of the quincunx, designed in 1889 by Sir Francis Galton, who famously coined the term eugenics and who also invented the correlation coefficient in his quest to prove that population distributions, including of intelligence, were a product of heredity, not environment. He designed his quincunx so that identical pellets poured through a funnel that randomly bounced off carefully placed pins would fall into different bins and produce a normal distribution, with the height of the column reflecting the probability of different pathways of descent. To Galton, this device beautifully demonstrated the properties of what he called, quote, the law of frequency of error, end quote, a law that he claimed, quote, would have been personified by the Greeks and deified if they had known of it. Each element, as it is sorted into place, finds, as it were, a preordained niche, accurately adapted to fit it, end quote. Now consider the alternative mechanical device shown on the left. It was built by several late 20th century physicists who altered the shape of the funnel and pins in order to generate a log normal curve, that is, a curve for whom the logarithm of the values has a normal distribution. Illustrating the interplay of structure and chance, it thus offers an illuminating mechanical metaphor for showing how altered structures can change population distributions, including of identical individuals, and do so by affecting the underlying probabilities of events randomly occurring to the population's individuals. Translated to thinking about population health, the implication is that rather than being determined by innate or fixed essence, the risk of individual occurrences of disease and death are inseparable from their societal and ecological context, a context which includes, among other things, both institutional and individual responses to both population rates of morbidity and mortality and well-being, and the magnitude of their health inequities. A useful analogy is to norms of reactions observed in individual organisms, whereby any given genotype can translate to different phenotypes depending on environmental conditions, albeit of course within the constraints of what is permitted by evolutionary history. A bee will not suddenly turn into a tree. The necessary historical corollary is that observed differences between populations are simply that, observed differences in a given context, not clear-cut indicators of innate difference. Structured chance allows us to see this possibility more clearly, and the structuring plays out over time and different spatial scales as well. How can we see this structure and make causal inferences? We can do so only by using theory. This is because theory is fundamental to scientific inquiry, such that any science concerned with explaining causal processes certainly needs to have some theory to guide inquiry and interpretation. More broadly, theory is essential because it provides both vision and insight. Indeed, traced to its Greek roots, the Greek word theoria refers to seeing inwards, such that to theorize is to use our mind's eye systematically, following articulated principles, to discern meaningful patterns among both ideas and observations. More formally, theory is key to elucidating coherent and presumptively testable sets of interrelated ideas that enable scientists independently to describe, explain, and predict features of a commonly shared biophysical reality in which cause and effect exists. Theory enables us to make invisible visible and to see gaps in constructs, variables, data, and their interpretation. Whether implicit or explicit, theory is inevitably at work in all of our public health work, so it is critical that we know what it is and what it entails. One useful metaphor for comprehending theory is John Zimon's map metaphor, presented in his last major work before he passed a few years ago, and it was titled Real Science, What It Is and What It Means. In the left-hand column, I list the conceptual and practical properties that Zimon argued maps and theories share as potentially useful, or I would add, potentially destructive, human endeavors to understand the features of domains of phenomena and the causal processes and connections they encompass and generate. According to Zimon, both maps and theories, first, always and necessarily have a viewpoint. Second, they focus on phenomena at specified scales, sometimes at specified level, other times engaging with cross-level connections. Third, they are meant to be useful, to get us from one place to another, literally, and also in our thinking and analysis. And as part of this, just about always inspire imagination, including, one hopes, about what is not on the map. And fourth, allow for what Zimon terms purposeful generalization. Fifth, they're always made by people in real context, that is, they are constructed, and they always are underdetermined, meaning that sixth, there can never be a map or theory of everything, since both are necessarily partial depictions and analysis of the presumed shared biophysical reality that I mentioned above. So that the necessary assumptions, these are the necessary operating assumptions for theories, maps, and even science to make sense. And so to illustrate his point, Zimon invoked, with words, the images I show here, 4 different maps of London, each depicting and answering different questions encompassing the same terrain. How to get around London, by car, by bus, by the underground, or by walking on the street. Where some might find only one of these maps to be of interest, others might want to engage with all 4 simultaneously, such as, for example, an urban planner seeking to make a city more ecologically sustainable or equitable in distribution of and access to resources, or someone in public health in analyzing in context how transportation and physical activity pertain to health. In keeping with this view of theory, epidemiologic theory, like any scientific theory, has a core set of defining features. These pertain to the domain the theory seeks to explain, which for epidemiology is population distributions of disease, disability, death, and health, and their determinants and deterrents across time and space. The objectives, in this case, to generate explanations of disease distribution relevant to improving population health, and most explicitly in the case of social epidemiology, reducing or eliminating health inequities. Third, substantive and explanatory concepts, which for epidemiology are about outcomes, exposures, populations, comparison groups, and causal pathways, each informed by metaphors that influence the kinds of causal mechanisms proposed. And because we're talking about research that involves populations and human agency, there is also, fourth, reflexivity, by which I mean that the explanation is intended to influence the phenomenon being explained, in this case, disease rates and the magnitude of health inequities. And fifth, historical contingency, since both the phenomenon to be explained and the explanations proposed, and who is proposing them, depend on societal context. As this list makes clear, epidemiologic theories of disease distribution are rich in content and cannot be reduced simply to theories of disease mechanisms, even as they are informed by them. Related, as in any scientific field, there can be contending as well as complementary theories. Thus, although there are commonalities across all epidemiologic theories of distribution regarding their domain, and also their use of substantive and explanatory concepts, there can be profound differences as well. Different theories can offer competing and different explanations of the same observed phenomena. They can have entirely different views on what they deem to be modifiable versus immutable, and they can have vastly different competing assumptions about who and what constitutes outcomes, exposures, population groups, and comparison groups. The net result could be very different causal inferences with very different understandings of what reflexivity entails, let alone attention to or disregard of historical contingency. How does this play out in contemporary epidemiology? Let me provide an overview of the different disease theories in use, after which I'll offer a very concrete application to show why choice of theory matters. On this slide, I list the leading dominant versus alternative epidemiologic theories of disease distribution informing contemporary research. The dominant frameworks are biomedical and lifestyle approaches. The alternative theories include diverse trends that generally cluster under the umbrella of social epidemiology and can, as I will describe, be subdivided into three general types: sociopolitical, psychosocial, and ecosocial. Of note, life course developmental theories can be incorporated into both the dominant and alternative perspectives, but how this is done depends on the theory at issue. We have already heard earlier this morning about conventional approaches or dominant approaches to causal thinking in modern epidemiology, so there is no need for me to review their theoretical underpinnings in detail. Let me just say instead that their dominant epidemiologic approaches, biomedical and lifestyle, tellingly are typically used implicitly rather than explicitly. What they share in common is first, a focus primarily on individual level pathology, biology, and behavior. Second, they emphasize factors amenable to intervention through the healthcare system. Third, they consider social determinants of disease to be at best secondary, if not irrelevant. Fourth, they view population simply as the sum of individuals, and population patterns of disease as simply reflective of individual cases. And fifth, they in effect reduce the whole simply to the sum of its parts. Dominant metaphors portray the body as a machine and/or the output of a genetic program, and choice is simply a matter of the taste of individual quote-unquote consumers, independent of context or constraints. Disease rates and distributions are thus conceptualized and analyzed as a consequence of ostensibly individual level innate biological phenomena and freely made choices, including those affecting the prenatal milieu, with history reduced to a question of individual biological development. Multiple causation is addressed through invocation of a web of causation, which is in effect spiderless, as the causal theory behind what and who appears in the web and what and who is left out is rarely, if ever, made explicit. By contrast, the social epidemiology frameworks that I list here are subdivided into three types for reasons I will explain. Although all distinct, nevertheless share four fundamental features not found in the dominant perspective. They are explicit about their theoretical premises, their view that the whole shapes properties of the parts, and vice versa, that is, the arrows go both ways, albeit not with equal force. They share a population orientation, and they have an emphasis that context matters. On this slide, I schematically show the commonalities and contrasts of the different variants of social epidemiologic theories of disease distribution. While each is concerned about the social determinants of health, they nevertheless conceptualize these determinants differently and differ in their focus on political economy, biology, and the kinds of causal pathways that they consider. Thus, the first major trend of the sociopolitical theories, rooted in the mid-19th century CE theorizing of Rudolf Virchow, Louis-René Viermet, and Frederick Engels, fundamentally posit that any given society's patterns of health and disease is produced, literally and metaphorically, by the structures, values, and priorities of its political and economic systems in conjunction with the society's technological level. Health inequities are thus posited to arise from whatever is each society's form of social inequalities, defined in relation to power, property, and privilege, both as internally produced and externally imposed. The theoretical attention is primarily on the political and economic determinants, and not the biological processes by which they are embodied. Examples of such socio-political theories include social production of disease, political economy of health, the many complex and wonderful different strands, theoretically, of Latin American social medicine and collective health, as we have just been fortunate to hear about from Jaime Breu, and we'll hear more later, and also fourth, the framework of health and human rights. Also germane are two increasingly depoliticized variants of these frameworks. First, what's called social determinants of health, and second, what gets called population health, both of which focus much more on consumption than production, and are much more social rather than socio-political in their orientation. By this, I mean that they lack or avoid explicit analysis of systemic, structural, economic, and political conflicts as determinants of population health, and focus instead on individual status within society. Stated another way, the health impact of low income is of concern, but why low income exists and who benefits from its existence is not. Reflecting this shift in orientation, their dominant metaphor no longer refers to production, but instead depicts quote-unquote upstream influences affecting quote-unquote downstream outcomes. For this reason, some epidemiologists, including myself, have begun to distinguish societal from social determinants of health, so as to ensure that structural determinants, and not only social rank and consumption, are addressed. The second major theoretical trend in social epidemiology in turn concerns the psychosocial determinants of health. The emphasis is on individuals' psychological perceptions of and their health-damaging or health-enhancing psychological, biological, and behavioral responses to social conditions, social interactions, and social status. The focus is chiefly on psychological and biological mechanisms, with less or no attention accorded to the socio-political determinants of psychosocial insults and buffers. The third set of socio-political theories is more ecological in orientation, and here I'm representing it by ecosocial theory, which I first proposed in 1994 and have elaborated since, as I will now explain. Centrally concerned with who and what drives current and changing patterns of social inequalities in health, ecosocial theory pays heed to societal and ecologic context, to life course and historical generation, to levels of analysis and to interrelationships between diverse forms of social inequality, including but not restricted to racism, class, and gender. A central focus is on embodiment, referring to how we literally embody biologically our lived experience, thereby creating population patterns of health and disease. Another is on accountability and agency, both for social inequalities in health and for the ways they are or are not monitored, analyzed, and addressed. With another key point being that the theory's core constructs are not intended to stand separately, but instead must be used together so as to promote integrated and systematic thinking. Let me now give an example that contrasts use of these different theories so as to set the stage for the more detailed application of ecosocial theory. The example I've chosen pertains to recent high rates of diabetes among the Pima Indians in Arizona, claiming to have the highest incidence in the world. On the slide, I show how each listed theory, excluding health and human rights because it's not yet been applied to this example, emphasizes markedly different determinants. At one end, the dominant biomedical and lifestyle account focuses on the so-called thrifty gene hypothesis, involving allegedly innate genetic differences selected for millennia ago in the Paleolithic era by allegedly periodic famines. A hypothesis that notably refuses to die, despite being discredited by both current genetic research and also historical research on famines, which are more prevalent under the post-Paleolithic systems of mass agriculture due to politically shaped distribution failures, not to a lack of food per se. And it also is of note that repudiated by its progenitor, James Neal, a geneticist, who at the end of his life rejected the idea there was any single quote-unquote diabetes gene, or that intrinsic genetic differences could explain higher rates of diabetes among indigenous populations now across the globe. Next, the population health approach, whose theorizing about thrifty phenotypes emphasizes pathways leading from a disadvantaged intrauterine environment to premature babies with disturbed metabolisms, and which, when combined with childhood and adult obesity, cause diabetes. And yet these theories have notably little to say about what has driven the actual timing and contours of the Pima's epidemic. Third, the psychosocial focus in turn is how stress affects physiology and health behaviors, leading to increased adiposity and hence higher risk of diabetes, but again offering few clues as to why the Pima's diabetes epidemic started relatively recently, given that stress has been around for a while. Fourth, the social production of disease or political economy of health focus on political and economic determinants of American Indians' living conditions, but with little explicit attention to the biological and ecological pathways, timeframes, and levels relevant to the actual onset and worsening of the epidemic. And finally, ecosocial theory, as further informed by North American indigenous health frameworks, supplements this last approach with its own added explicit clarity about the importance of political ecology, spatio-temporal scale, and life course in relation to historical generation and place. Thus, in the case of the Pima, at issue is what it meant to have been forced on reservations in the late 19th century, the ensuing famines, followed by their main source of water being diverted for use by white farmers and cities such as Phoenix, Arizona, in conjunction with the post-World War II U.S. government provision of high-fat, low-nutrition, quote-unquote, commodity foods, and what it means now, 80 or so years later, for the Pima to begin to regain control by litigation of their water and hence start to recover food sovereignty. This approach explicitly engages with biology by shifting attention from gene frequency to gene expression, positing that any people subjected to the same history and conditions would likely have had a rapid rise in diabetes rate, as has been seen in different indigenous populations who have very different population ancestries around the world. And with this trend potentially exacerbated by transgenerational biological amplification likely occurring in utero, given how maternal obesity during pregnancy appears to have an epigenetic influence on insulin efficiency and metabolism of both the fetus and subsequent infant, assuming that it is live-born and survives. In other words, choice of theory matters for explanations and hence remedies as well. So let me now give a bit more depth in ecosocial theory. I first began sketching out this theory in 1994 in an article titled Epidemiology and the Web of Causation, Has Anyone Seen the Spider? A title that came to me in the midst of a rather vigorous argument in an epidemiology seminar, where the web of causation and its endless array of so-called proximal and distal risk factors was being praised to the skies. And in a moment of frustration when trying to push the discussion in the direction of agency and accountability for the presence or absence of these factors and their connections, I asked, has anyone seen the spider? The metaphor of the spiderless web of causation set thoughts in motion, and I soon worked my way beyond thinking just about spiders to thinking about how to take seriously the fractal intertwining of the societal and biological at each and every level, from macro to micro. Hence the proposed metaphor used by ecosocial theory of the tree or bush of life jointly intertwined with the scaffolding of society, one constantly built up and altered and contested and developed by people in ways that can permit the biological either to wilt or to flourish, which in turn has implications for the sorts of societies people build. And it also has implications for how we study population distributions of health, disease, and well-being, as I will now explain. So let me give you now a more in-depth example of the application of ecosocial theory. My basic thesis is that how we think about biology in historical, ecological, and societal context matters for framing causes of and solutions to health inequities. The contrast I offer is between dominant gene-centric approaches that treat disease and biomarkers as inherent characteristics of individuals and population groups versus critical approaches such as ecosocial theory and also the critical epidemiology that we have just heard about that takes history seriously and views biomarkers, disease rates, and disease inequities instead as embodied history, that is, embodied phenotypes. More specifically, for well over a century, the dominant paradigm has placed genes and gene frequency at the center of biological phenomena, including disease and disease rates. Reflecting this orientation, funding priorities for the U.S. National Institutes of Health would emphasize genomics, including in relation to social inequalities in health. Indeed, in the NIH's most recent biennial 732-page report to the U.S. Congress, within the 46 pages on minority health and health disparities, the terms genomic, genome, genetic, and gene appear 87 times, whereas social determinants of health and discrimination each occur once, socioeconomic 7 times, poverty twice, racism not at all. During the past 2 decades, however, a fast-emerging body of biological research is challenging the dominant gene-centric paradigm with robust, novel evidence that nothing in biology makes sense except in the light of history: evolutionary, developmental, ecological, and societal. Termed ecological, evolutionary, developmental biology, or ECO-EVO-DEVO for short, a central thesis building on ideas of norms of reaction is that, quote, the same genotype can generate different phenotypes depending on what cues are present in the environment, end quote. Hence, quote, bodies express ecology, end quote. Providing novel evidence of environments not only select or filter variation, but actually construct it, well-known textbook examples of non-human illustrations of developmental plasticity in embryos and phenotypic flexibility in adults range from temperature-dependent sex determination in turtles to socially induced sex change in adult fish, to massive changes in adult migratory birds, gizzard size and muscle type, depending on where they are in the course of their migratory cycle. Among humans, one very salient example is the differential adult health status of identical twins reared together, but with divergent adult social class trajectories. What relevance, if any, might this broader, more historical, and more dynamic framing of biology, only just now beginning to make headway into the public health literature, have for understanding and addressing health inequities? The approach I will take is to consider critically here using the example of the breast cancer estrogen receptor, its 4 histories. History 1 pertains to health inequities, the second history to individual or life course, the third history to cellular pathology and the natural or really unnatural history of disease, and the fourth history that pertaining to evolution. As for why the breast cancer estrogen receptor matters, it's a biological trait of critical clinical and public health importance because women and men whose tumors are ER positive can be treated with anti-estrogenic drugs such as tamoxifen and meloxifene, and hence have better survival than patients whose tumors are ER negative. Of note, the estrogen receptor is not exclusive to breast tissue. Present in people and other vertebrates, it's a steroid receptor, chiefly located in the nucleus, but also in cell membranes, that in its 2 different forms, ER alpha and beta, is centrally involved in regulation of cell growth and of cell death throughout the body, including the reproductive, immune, skeletal, cardiovascular, and central nervous system. A defining feature is that the estrogen receptor, like any steroid receptor, has the dual ability to bind DNA regulatory elements while at the same time having its own activity be modulated by substances which attach to it, that is, the ligands. What then is the conventional view of epidemiology of the population distribution of the breast cancer estrogen receptor? In brief, reflecting conventional theories, most studies on breast cancer ER status, like other mainstream research on risk factors for disease, typically investigate risk factors for breast cancer ER status ahistorically, as if it were a fixed trait of tumors and individuals. Identified risk factors for ER-positive tumors are stated to be late age at menarche, delayed age at first birth, higher parity, breastfeeding, postmenopausal obesity, and use of hormone therapy, especially estrogen plus progestin. For ER-negative tumors, they are premenopausal or younger age diagnosis, and also being a carrier for BRCA1, which is a harmful mutation of the breast cancer susceptibility gene 1. In US research especially, but not exclusively, race is also listed, whereby ER-positive tumors are stated to occur more frequently among white women of European ancestry as compared to women of African and Asian descent, a difference typically inferred to be genetically driven. Underpinning such inferences is the long-standing but erroneous belief that humanity could be divided into biologically discrete races, a stance still explicitly argued by some researchers, even though not supported by contemporary evidence. Thus, in the sweeping words of one typical review article, quote, women of African ancestry tend to develop early onset, high-grade, and ER-negative tumors more frequently than women of European ancestry. Quote, early onset ER-negative tumors also develop more frequently in Asian, Indian, and Pakistani women, and in women from other parts of Asia, although not as prevalent as it is in West Africa. These are rather huge regions of the world. And also, quote, populations with low socioeconomic status are more likely to develop an ER-negative disease than populations with a high SES. Also emphasized in the global literature, albeit based on incredibly scanty data given the lack of cancer registries in poorer countries, is the on-average younger age diagnosis associated with more aggressive ER-negative disease among women in what are called, quote-unquote, developing as compared to, quote-unquote, developed countries. Does, however, an historically informed analysis support these sweeping assertions and their underlying causal premises? Let's look first at the history at the societal level, comparing trends in the rates of and inequities in cause-specific mortality and morbidity. Such information has long proven critical to understanding determinants of both population health and health inequities, even as is it all too often overlooked in the rush for the next latest discovery and dismissed as merely descriptive. Yet it is these data that reveal observed disparities in ER status are historically contingent, not fixed. Jump back first to the middle third of the 20th century CI, a time predating both discovery of the estrogen receptor and its clinical use in guiding breast cancer treatment. This period also precedes the widespread rise of mammographic screening and hormone therapy. It consequently was a time when breast cancer incidence and mortality rates more closely resembled each other as compared to their divergence now due to changes in both screening and treatment. As shown in the table on the slide, contrary to current findings, premenopausal breast cancer mortality rates indicative of early onset disease, more likely ER negative, were in 1931 lower and with a less steep age-related increase among women in the UK who were married to men in working class as compared to professional occupations, with this only shifting by 1971 to where these rates were nearly equivalent. Similarly, in the US, premenopausal breast cancer mortality rates in the 1930s were virtually the same for white women and women of color, a pattern that continued well into the 1960s. Moreover, in both countries, the proportion of breast cancer deaths under age 50 in the 1930s was also far higher observed than is today, and that would be expected given the shorter life expectancy back then. Although such data must be interpreted cautiously, given possible misclassification, likely differential, of cause of and age of death, social class, and race ethnicity, the most likely bias involves census undercount of more impoverished persons and people of color, shrinking their denominators. Consequently, the data in the table shown, if anything, present an inflated estimate of risk among women subject to social adversity, suggesting even less resemblance to contemporary patterns. Further challenging ahistorical approaches to analyzing and interpreting racial, ethnic, and social class disparities in ER status are results of a study that my team and I recently published, the first to test statistically trends in the white-black odds ratio for ER-positive breast cancer, examining women diagnosed between 1992 and 2005 with incident invasive disease. Using data from the 13 US major cancer registries, we found that the age-adjusted odds ratio rose from 1992 to 2002 and then leveled off and actually fell among women aged 50 to 69. The most plausible explanation is that these results reflect the socially patterned abrupt decline in hormone therapy use following the July 2002 publication of results of the Women's Health Initiative, which found that hormone therapy did not protect against and may have elevated risk of cardiovascular disease, and at the same time reconfirmed prior evidence that hormone therapy increased risk of breast cancer, especially ER-positive breast cancer. As a result, hormone therapy use dropped precipitously among the women most likely to use it, which in the US were white, affluent women with health insurance who were sufficiently healthy not to have any contraindications against use. They accordingly experienced a drop in the prevalence of ER-positive tumors not observed among women who did not use hormone therapy, thus reducing the white-black odds of ER-positive tumors. Hence, as these two slides should make clear, current social inequalities in ER status are precisely that, current, not invariant. Contemporary findings should always be evaluated in light of historical trends. Now, comparing risk across historical generations provides one kind of insight. Comparing risk over the course of individuals' life course, that is, their individual history, offers another. Here, the case of women with repeat occurrences of breast cancer is particularly instructive, especially because all exposures occurring prior to the first tumor are common to both tumors. Of note, since at least the mid-1980s, studies have repeatedly documented considerable discordance in ER status, independent of treatment status, among women with repeat diagnoses of breast cancer, both tumors arising from metastases and those occurring as new primary malignancies. For example, recent evidence from large US and international studies indicate that women whose initial primary tumor was ER positive are equally likely to have their second primary tumor be ER positive or negative. Moreover, among BRCA1 and 2 carriers, discordance of the ER status between first and second tumor is, for women who are BRCA1 carriers, as high as 70% for those whose initial tumor was ER positive, and for women with BRCA2, as high as 48% for women whose initial tumor was ER negative. The finding that new secondary primary tumors and even metastatic tumors can differ in ER expression, even among BRCA1 and BRCA2 carriers, underscores that ER status cannot be construed as a fixed trait of individuals. So a third history pertains to that of the tumor itself, or more broadly, cellular pathology. At issue is the well-known, quote-unquote, natural history of disease, recognizing, of course, that disease is not simply natural, but can vary by individual societal, ecological context in terms of its course. Also, for breast cancer, like many diseases, there is not simply, quote-unquote, one disease. Instead, there are a variety of types, heterogeneous at the molecular as well as clinical and epidemiologic levels of analysis. Thus, as shown by the figures on this slide, which come from the work of Cornelia Polyak, breast tumors can be distinguished by not only such well-known clinically relevant features, such as whether they are positive or negative for ER receptor, also progesterone receptor and other receptors, but also reflecting new technological advances by molecular subtypes placed on global gene expression. Current work on clonal competition, moreover, clarifies that it is erroneous to assume that a tumor is of only one type. Instead, as increasingly emphasized in clinical oncology research, intertumor heterogeneity, whether for the estrogen status or at the molecular level, changes over time. New experimental evidence additionally suggests that ER-negative tumors, once thought to be permanently ER-negative, can be induced to re-express the estrogen receptor. Although the idea of tumor as emergent phenotype has appeared in the carcinogenesis literature for at least half a century, it nevertheless is largely absent from public health research regarding etiology, which continues to treat ER status as fixed, as opposed to reflecting whatever the tumor predominantly happens to be when it is detected and biopsied. As the evidence shows, however, the history of a tumor, and hence that of its constituent cells, matters, not only for treatment and prognosis, but also for etiologic analysis and inference. Lastly, consider the contemporary relevance of the evolutionary history of the estrogen receptor and other nuclear receptors. A topic of lively investigation and debate, new evidence amassed in the past decade, as summarized by the genealogy from Baker, shown on the upper right hand of the slide, indicates that nuclear receptors evolved about 635 million years ago, long before any organisms had endocrine systems, and they are ubiquitous in animals, but not present in fungi, plants, or choanoflagellates. The evidence further suggests that the postulated ancestral estrogen-related receptor, also referred to as the ancestral steroid receptor, gave rise to the vertebrate estrogen receptor, which uniquely has the ability to bind estradiol. Moreover, as clarified by the additional genealogy shown on the bottom right from Ike and Thornton, only vertebrates have the two forms of estrogen receptor thus found in the human breast, ER alpha and beta. One salient leading hypothesis, shown by the figure from Sladek on the left hand of the slide, is that the ancestral estrogen-related receptor, for which no steroid ligand had yet evolved, initially functioned as a xenobiotic sensor, detecting substances exogenous to the organism that could be eaten or else require detoxification. Another leading hypothesis is that subsequent diversification of steroids and nuclear steroid receptors in vertebrates enabled the occurrence and control of complex physiological processes during the development and adult life of vertebrate organisms, including our hormonally regulated reproductive system, which for mammals includes breasts, and hence our generic name of Mammalia. And as for the relevance of this history to the breast cancer estrogen receptor, it is expressed in the estrogen receptor's evolved sensitivity to extracellular signals. Thus, estrogen receptor expression, far from being fixed, can change over time repeatedly. Altering expression can be shifting hormonal levels, whether endogenous, as in the case of pregnancy, or endogenous, as per exposure to hormone therapy, and also other exposures. Estrogen receptor status is thus a flexible characteristic of cells. Vertebrate cells' evolved capacity is to change their estrogen receptor status, which consequently is contingent, not intrinsic. The evolutionary history of the estrogen receptor helps explain why this flexibility exists. In conclusion, as this case of the estrogen receptor for breast cancer reveals, history matters deeply at multiple levels and timescales to claims about disease, etiology, and causes of health inequities. In particular, these findings challenge static and gene-centric approaches to biology and etiology, including approaches that treat measured biomarkers as primarily reflections of innate biology and observed inequities in their distributions as fixed. The new work in ecological evolutionary developmental biology is providing compelling organisms that phenotypes are emergent and flexible. So the four histories considered here, societal, individual, pathological, and evolutionary, and the levels that they span are at play in every case and every population rate of disease. They are concurrent, not sequential. On the slide, I accordingly list four sets of historically informed ecosocial questions based on conceptualizing health and disease as emergent embodied phenotype that I propose could be asked for any health outcome, with the point being not to presume the answers, but to find out empirically what the evidence shows. The first asks us to consider what data exist on historical trends in the on-average levels or population rates of, and the health inequities in, the embodied biomarker